Hi, we'll wait for a few minutes so that others can join, and then we'll start with discussing the assignment problems for this time. Okay,
Okay.
thanks. Thanks. Hi, am I audible?
Yeah, yeah.
Yeah, so I think we'll start now, and slowly as students join, they can pick up and we can repeat if needed. I'll open my presentation now. Yeah, can you see my screen? Can you see my screen?
Yeah.
Yeah, okay. Um, yeah, good evening. So, for today's discussion, we'll be looking at a few sample assignment problems that are related to the lectures that were posted for week 2 of this NPTEL course, Metabolic Engineering. I'm Shankar Mahesh. I'm a PhD student from IISc Bangalore. Surinder, I had checked the discussion forum. You had posted a couple of questions regarding lecture 6. We'll be discussing that as well in this uh, discussion session, and based on that we can also have an answer posted later in the discussion forum. That should be fine, right?
Okay.
Yeah.
Okay.
Oh, okay, cool. Okay, so for this week's course, what I could understand from the lectures is that it was mostly related to the metabolic pathways within the cell, which is like the pathways that convert sugars, the three different pathways, and transport system pathways. And one of these also had answered a question that we had previously discussed, which was about the difference between facilitated diffusion, if it is active or passive. So we'll discuss that as well, Surendra. This was also a question posed by you last week. So we'll discuss that as well. So
Okay.
to start with, we'll look at these questions. As usual, we can have you can start raising your hand as you read a question and try to justify why you think a particular answer could be the right answer. You can give a reason, and then we'll discuss and then we'll finalize. So the first question is, which of the following options are incorrect for the precursor molecules which provide the resources for the biosynthesis of large number of macromolecules? The options are: option A, metabolites with high molecular weight; option B, they are rapidly used for biosynthesis of macromolecules; option C, they are derived from glucose or other carbon sources; option D, none of the above. You can take a couple of minutes and then raise your hand. Yeah, Surendra, you can start by talking about one specific option, then we'll give Ayush a chance.
Okay. So, precursor molecules, they are used for the synthesis of the macromolecules like
Huh,
proteins, lipids,
huh,
and these precursor molecules are small molecular weight, low molecular weight compounds
okay,
that are derived by the carbon metabolism.
okay.
Okay, so when
So,
cell take the glucose, it
huh,
metabolized to
huh,
glucose 6-phosphate and ultimately to pyruvate.
huh,
So glucose 6-phosphate, pyruvate, they are the precursor molecules. They are used in the various biosynthetic pathways.
okay.
So and rapidly they are used, not used. If we are having a more precursor molecule, it doesn't mean biosynthesis of the macromolecules will be more because macromolecules are synthesized according to the requirement of the cells.
No, no, no.
So that's
That
why.
that option is not to be understood that way. When they say rapidly used for biosynthesis, it means if it is immediately consumed. They don't say that just because a lot of precursor is available, a lot of biomass is accumulated. They don't say it that way. That is the topic, but that is not covered in this particular question.
Okay, so I go with the C option,
Okay, so
derived from the
yes.
glucose and carbon. So,
Okay, the question is which of the options are incorrect? Ayush,
incorrect. Oh, sorry.
yeah, yeah. Which of the options are incorrect?
Uh, okay. It is A.
Okay, Ayush. Uh,
Yes, sir. It's a because precursor molecule doesn't precursor metabolic molecules doesn't have higher molecular weight. Uh,
okay,
Answer C or B, sir, are generally the correct answer because
right.
the precursor is generally derived from glucose and other carbon sources as just it breaks down and form the smaller precursor molecules.
Huh, right. Like both of you had discussed, the answer is option A because metabolites, these precursor molecules, have lower molecular weight. True. Moving on to the second question. Uh, why is it often necessary to make large number of genetic modifications to alter cell metabolism? We can take a couple of minutes. Then each of you can take turns and try to say why a particular option is either right or wrong. Yeah, Surinder. So you can start. Yeah, Surinder. So you can start with option A. Why do you think if it is right or wrong? You can just tell that. You need not tell the right answer.
Because if one metabolite can be synthesized by the many pathways, That's why I think multiple modifications are required to alter these pathways. A single molecule can be synthesized by the multiple pathways. Moreover, competitive pathway we have to block also. We have to block the competitive pathway also.
Yeah, yeah, that is in relation. But the question is, like, why it is necessary to make many genetic modifications? So we have a few options. Like the first option is it is difficult to identify and modify the pathways. The second option is because there are large number of control mechanisms within the metabolic processes. C is utilization of the precursor molecules to synthesize macromolecules takes prolonged time. And option D is large numbers of metabolic pathways are involved that lead to the production of a number of precursor molecules from nutrient sources. What I would want from you is why do you think option A is either right or wrong? So you can
It
just,
is wrong.
okay, why do you think it is wrong that it is difficult to identify and modify the pathways?
Uh, because nowadays advancement has been done so much, na? That's why I think it is not so much difficult task to identify the pathway. Carbon dating can be done easily.
Huh. So to just
The
rephrase
pathway
what
can
you
be
had,
identified.
so to just rephrase what you had said, because the organ it is now easier relatively to sequence the organism and get the genome of the organism. One would be able to get the genome of any cell, and when you get the genome you get the protein coding genes and information as such. And when you're able to map that, it is now easy to identify and be able to connect pathways, so that way you'll be able to modify pathways. So that is not necessarily the right reason why one has to make large number of genetic modifications. Ayush, what do you think about option B? Do you think it's right or wrong? You can give an explanation.
Yes, sir. I think it's right because sir, generally, as we know, uh, any cell or any microorganism that we are working to uh, to produce any product.
Huh.
So it has a large number of uh, large number of genes that are producing the same gene or producing the same generally responsible for the same protein or a different type of protein. So
Huh, Ayush, I can hear you.
uh, sir, we have to make a specific uh, gene that are encoding a uh, specific metabolite, so we can able to extract the uh, uh, sir, I'm not confirm about that.
Okay, I can explain that. So, as we know, in a metabolic pathway within an organism, the genes, the proteins, they are all interconnected, and one metabolite is either produced by many pathways or also taken by many pathways. So, in order for the synthesis to be uh, controlled, we have different mechanisms that uh, has a checkpoint kind of at every step, and that determines whether a particular pathway has to be controlled. So, if we take any particular metabolic pathway, because it has many different control mechanisms that decides whether the genes involved in the pathway are being expressed at that optimal level at that optimal time, it means that we have many different mechanisms that have to be taken into control to make the genetic modifications. So it is actually true that there are a large number of control mechanisms within the metabolic processes that make it difficult for cell metabolism to be engineered, making large number of genetic modifications. What you said is along the same lines, I think.
Yes, sir.
And option C is that utilization of the precursor molecule to synthesize macromolecules takes prolonged time. Surinder, you can share your opinion on this, whether this is right or not.
Uh, Actually, in my opinion, this is incorrect
Huh.
because I studied that E. coli cell can make a protein in 20 minutes. Okay, that's why I say making protein in 20 minutes. It is a faster process. E. coli they make their proteins in a faster way. Don't take much time.
Huh. Okay, just to make one clarification, E. coli cells themselves can divide in under 20 minutes.
Mhm.
So
Yeah,
the
yeah, yeah.
synthesis of proteins are happen in less than seconds, less than a few seconds.
Mhm. Yeah,
So
yeah, yeah.
technically, what they have mentioned in option C, that the utilization of precursor molecule to synthesize macromolecule takes prolonged time, is wrong. And option D, which is large numbers of metabolic pathways are involved that lead to the production of a number of precursor metabolites from the nutrient sources. So this one also, I think it is right. Yeah, I think this is right. Large numbers of metabolic pathways are involved that lead to the production of a number of precursor molecules from
Mhm.
nutrient sources. Just like how we know that there are many control mechanisms for one particular metabolite to be synthesized, similarly, there are many mechanisms as well to synthesize. Only when there are many pathways to synthesize, there will be many control mechanisms to prevent that synthesis or that or enhance that synthesis. So the right option for this particular question is option E, which is both B and D. Because to produce a particular molecule, we have many different pathways, and many different pathways have many different ways to control the pathways. It makes it difficult to engineer the pathway within the cell, requiring many number of genetic modifications. I hope that was clear.
Yeah, yeah.
Yeah.
Can you explain A again?
Yeah. Yeah.
Why A is incorrect?
Yeah. 1 minute.
A option.
Yeah. 1 minute. So what they say is it is difficult to identify and modify pathways. And the question is, why is it often necessary to make large number of genetic modifications to alter the cell? So, like you had mentioned earlier, like currently we have enough resources to be able to say, to be able
Mhm.
to talk about pathways. Like, I can give you one example. If you say you have the genome of an organism, from the genome itself, you can create a model of the organism computationally called as a genome scale model. So this genome scale model would account for all the reactions, all the metabolites, and all the intermediates within the cell. So this means the entire cell's metabolome and reactome has been mapped,
Mhm.
making the identification and modification of pathways easier. In the further lectures, we'll be talking about a method called flux balance analysis. In those flux balance analysis, what we do is we take the genome, make a model called the genome scale model, and try to mathematically compute how to distribute flux. So it is not difficult to identify and modify the pathways, which is why this option is incorrect.
Okay.
Ha. The next question is central carbon metabolism enables the cells to produce option A precursor molecules, option B macromolecules, option C A and B, option D none of the above. So, excuse me. Uh, yeah, Ayush, you can start with option A if you think option A is right or wrong.
So,
Yeah, sorry.
so uh, as we know that the
Sorry, sorry. You can go ahead, Ayush.
sir, I think sir A should be the answer, sir.
No, I just wanted to know from your side, why do you think option A is right or wrong? So you can try to
Sir, I'm not
explain
able to
that.
hear you.
Oh, sorry. I just wanted to know why do you think option A is right or wrong? You can give an explanation, then we'll finalize. Kritika, can you keep your mic on mute? We'll hear your opinion next. Yeah, Ayush, you can say. Why do you think option A, which is precursor molecules, is right or wrong?
Sir, because generally, central carbon metabolism provide the cell with essential precursor molecule. Like example in acetyl CoA,
Huh.
the central intermediate of that feed the pathway leading to the synthesis of fatty acid and the amino acid.
Hmm. Okay, so you think that the central carbon metabolism enables the cells to produce precursor molecules.
Yes, sir.
Okay, we'll discuss further. Uh, Kritika, do you think macromolecules are also produced by uh, due to central carbon metabolism? You can give your opinion.
So, basically, central carbon metabolism helps in production of precursor molecules,
Yeah.
and these molecules can be used to produce the amino acids which are required in proteins and in some cases nucleotide subunits as well. So, I think both A and B can be correct because indirectly they are producing the macromolecules. I'm not sure about the second option, but first one is correct, sir.
Okay. Surinder, would you want to say if options both A and B are right, or do you have a difference in opinion?
It is A is the correct,
Okay.
because central carbon metabolism includes the glycolysis and TC cycle.
Yeah.
So by the process of glycolysis and TC cycle, many metabolites are produced that are called the small metabolites, means precursor molecules
Precursor molecules.
like pyruvate, acetyl CoA,
Yeah, there are
hydroxy.
12 precursor metabolites, including glucose
Yeah, 12.
6-phosphate, fructose 6-phosphate, alpha
So these
ketoglutarate.
precursors are
Yeah,
used for the synthesis of the macromolecules.
true. So, in a way, central carbon metabolism enables the cells to produce both precursor metabolites and macromolecules. So, option C is actually the right answer because without central carbon metabolism, you'll not have precursor molecules, and without precursor molecules, you'll not have macromolecules. And since both options are here, we can surely say that both options are right.
Okay.
Yeah. Kritika and Ayush, was it clear?
But it is
Yeah.
asking na central carbon
Yes, sir.
metabolism
Yeah. It is
enables.
asking,
If I
it
ask
enables the
what is the
cells to
product,
produce, they don't, that way, central carbon, yeah. So that way, central carbon metabolism also contributes to biomass as well. But among the given options, since both of these, which is precursor molecules and macromolecules, indirectly are being produced by central carbon metabolism, the right answer is option C. Uh, should I repeat, Surendra, or was it clear? Or
no, no, it is clear.
okay, okay. So the thing is, they Because they've asked macromolecules, and because macromolecules are produced from precursor molecules, we can consider both as the right answer. That's why we consider it this way. Uh, the next question is synthesis and organization of macromolecules are required for option A, cell growth, option B, maintaining homeostasis, option C, maintaining functions. So uh, we can take a couple of minutes and then each one of you can try to justify why one of the options is right or wrong. Yeah. So who wants to go first? Yeah, Kritika. You can talk about option A. Why do you think option A is either right or wrong?
Sir, according to me, all options are correct because macromolecules, that is protein, nucleic acids, so all these are required for the cell growth. When we talk about the cell cycle, enzymes are required in them. Those are proteins. If they will not form in the enzymes which are required in cell cycle, those then this process will not occur.
Okay, so you're saying macromolecules are required for cell growth. Yeah, Surinder, you can talk about why, you can share your opinion on why macromolecules are required for maintaining homeostasis.
Homeostasis means balancing.
Yeah,
Okay.
maintaining
So, I mean,
the cell's normal state in which it
yeah, normal state.
exists. Yeah.
Yeah. For this, cell has to take the molecules from its environment. Also, cell has to excrete the waste material
Huh.
outside the cell.
Yeah.
Okay. So, for the transport outside and inside, some proteins are required. Macromolecules are required.
Yes.
Also, maintenance of the calcium balance inside the cytoplasm. Okay, it also required the proteins.
Huh.
So that's why I think it is a correct. B is the correct.
Right.
Means all are.
Sorry, can you repeat? That was not clear.
According to me, B is also correct. Means
B is also
all of the above.
all
Uh,
of the above.
B,
Okay.
all of the
Okay.
above.
Huh. Ayush, do you think option C is right?
Yes, sir.
Okay.
So because protein serve as enzyme and the structural component or receptor transporter and play generally play the key role in the cellular activities. So maintenance of the functions is also correct.
Right. Yeah. Like all of you had mentioned. This synthesis and organization of macromolecules are required for all these three options, which include cell growth, maintaining homeostasis, and maintenance of functions. So, cell growth talks about division of cell, meaning requiring proteins that are involved in cell division so that partition of the cell's resources is done from mother to the daughter cell. Then, once the cell divides, it has to maintain its states, the state in which it is present, which depends on the function, which determines what all roles the cell's organelles has to play. So all these macromolecules are required for all the three different options that are shown here. So the right option is option D. Moving on to the next question. Uh, Presence of more amount of precursor molecules necessarily means higher level of biosynthesis that will be occurring naturally. So uh, you can take a couple of minutes and yeah, Kritika has raised her hand. You can, instead of telling the right answer, Kritika, you can this time try to say uh,
I have to
what,
give an explanation.
yeah, what exactly happens given we have a particular amount of precursor molecule. You can take a couple of minutes and then I'll tell you and then you can start.
Okay, sir.
Yeah, Kritika, you can unmute and talk.
Yes, sir. So, like what I've studied through the lectures, so it was mentioned there that a small amount of precursor can leads to the biosynthesis of the molecules. So it doesn't mean that if we increase the precursor that the it will cause a synthesis of macromolecules or the things we required through this procedure because like as we have said other conditions are also required for the synthesis, so it depends on the other factors as well.
Yeah, do you want to explain what you mean by these other factors if you're aware?
So like when we talk about, let's take an example of glycolysis.
Hmm.
So when we talk about the phosphofructokinase, we will
Okay.
take that step of this reaction. So we will see that there is condition when there is ATP in large amount that will inhibit the phosphofructokinase while AMP increase its efficiency. So these conditions can affect this biosynthesis.
Okay. We will comment on it after listening to others. Surender and Ayush. Yeah, Surender. You can
Uh,
give your opinion without specifying what is the right answer.
uh, in our cell, in our body, or in E. coli body,
Yeah.
uh, amount of the macromolecules
Huh.
they require for a particular function,
Huh.
and optimum amount of the macromolecules are synthesized also. If some macromolecules is no more required
Hmm.
because it is of no use now,
Huh.
cell function has been finished. Cell function has been finished,
Huh.
so protein is no more required.
Right.
So automatically, synthesis of that particular macromolecule from the precursor molecule will be inhibited.
Right.
For example, if we are having more acetyl-CoA, so it will inhibit the PDH complex. That no more acetyl-CoA is required. It is already sufficient amount of acetyl-CoA is there. So excess acetyl-CoA is not going to synthesize the macromolecule. Instead, excess acetyl-CoA they will inhibit the its production. They will inhibit their production from PDH complex.
Right. Right. So what you mentioned is an idea about the control mechanisms that exist that prevent from random utilization of precursor molecules. Good.
Yeah, yeah.
Ah, Ayush?
Yes, sir.
Yeah. So, what do you think about this particular question? Do you think if we have more of precursor, we'll get more of biosynthesis of large macromolecules, or will there be some kind of regulation, or what do you think will happen?
Sir, 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 it can be, it cannot be, sir, because as the earlier said by Kritika and the so in the that all the process have their any a confirm a fix uh, a fixed reaction or the the inhibitors that can hello sir.
Yeah, yeah, we can hear you, Ayush. Yeah.
So, as we said, that all the reaction have been in the fixed amount and the fixed manner, so that there always be chances of that the having the more precursor will lead through the even though it's lead to the production of the production of the higher molecule high level of biosynthesis, but it will stop by the sir,
Huh. Yeah, we can hear you. You can give it a try, or I can explain it,
sir,
taking
you
from
can explain that.
Kritika's and Surendra's answers.
Yes, sir.
So one thing we can understand from the cell is that it does not always produce all the compounds, or all it does not basically synthesize all the metabolites that it can synthesize at the same time. There is synthesis happening only when there is requirement. Meaning, even if you have a few millimoles of a particular precursor available within the cell, unless there is a macromolecule which needs to be synthesized for a particular function which requires this precursor, then this precursor will not be utilized. So, this is what Kritika had said, and Surender had talked about control mechanisms that exist which decide whether a particular precursor will be taken up by a metabolite or not. So one could also think of enzymatic inhibition, which is caused when a large amount of a particular substrate is present. Like enzymes are proteins that have particular substrates. In certain cases, what happens is the same enzyme which binds to the same substrate that binds to the enzyme at a very, very higher concentration prevents the enzyme from functioning. So this is a natural regulation mechanism that naturally happens within the cell that prevents the precursor molecules to be fully exhausted. So these are control checkpoints, and the right answer is true. Uh, that the presence of more amount of precursor, sorry, the right answer is false. The presence of more amount of precursor molecules necessarily means higher level of biosynthesis that will be occurring naturally. It is false. Just because we have more precursors, it doesn't mean that we'll have a lot of biosynthesis happening. It only depends on the function of the cell, which depends on the state of the cell. I hope that was clear. This is also given in lecture 6 at around 24 minutes, where they talk about tight regulation and the controlled use of precursors for the biosynthesis of amino acids, lipids, nucleotides, and carbohydrates. So, if there are any doubts, this lecture 6 can be looked at. It comes at around 24 minutes. Okay. The next question is, enzymes elevate the activation energy, which is necessary to transform a reactant into a product. So, we can take a couple of minutes and then, Kritika, you can try to say what activation energy is, followed by which we'll discuss the right answer. Yeah, Kritika, you can start by explaining what activation energy is, and then we'll go ahead with Surrender, and then Ayush. Kritika, are you there?
Yes, sir. Can
Yeah,
you hear me, sir?
yeah, I can hear you. We can hear you.
Okay, sir. So basically, sir, actually every reaction have a threshold value, which it should be, which it should cross to take place a chemical reaction. So activation energy is defined as that minimum energy which is required to further take the chemical reaction to proceed. And sir, can I give a further information like what happens when enzyme is present and what is?
Uh, we'll we'll come for that. So
Okay,
what you're saying about activation energy is that it is the minimum
minimum
energy
threshold.
that needs to be supplied. So
Yes,
any reaction takes place when there is a breakage of bonds or addition of some chemical moiety to some other chemical moiety, which also
yes,
involves breaking and making of bonds. And breaking and making of bonds involves energy. So activation energy can be correlated to the amount of energy needed to make or break the bonds involved in a particular reaction. Uh, is that a fair statement to make?
Yes, sir.
Okay. Uh, yeah, Surender. So you can talk about what happens in the when enzymes are not present to the activation energy of a particular reactant to convert it into a product. In a
Can you
case
repeat the
where
question?
enzymes are, yeah. So let's say you know the enzyme activation energy of a particular conversion from a reactant into a product is X in the
Mhm.
absence of the enzyme. So how does an enzyme specifically uh, change this X into Y? What an enzyme actually does to the activation energy.
Okay. So, actually, for example, I take the example of a sticker. If, for example, I have to break the stick into two pieces.
Right,
Okay. So I have to first bend it.
right.
Okay. Means straight stick to the bended stick. So it will take some energy. From straight stick will go to the bended state. It will take energy. That is the activation energy.
Haha, that's the activation energy. Yeah.
Okay.
Huh.
Activation energy. We have to bend it, then it will break
Right,
into products.
right. So what does enzyme specifically do to the activation energy?
They lower the activation energy. Means less energy required to bend the stick.
So the question is whether enzymes elevate the activation energy or not.
No.
So the answer you are saying is it basically lowers the activation energy.
Lower the activation.
Okay. Huh. Ayush,
Yes, sir. Sir, it lowers the
so
activation energy, sir.
it lowers the activation energy. Okay. So to just give a brief conclusion, when two reactants interact, they attain a state called transition state. For example, when an enzyme interacts with the substrate, it forms an enzyme substrate intermediate. And what happens here is the amount of energy, which is represented in Gibbs free energy delta G, is usually different for the enzyme substrate complex rather than the substrate alone. So because of this difference in the delta G value, in the presence of the enzyme, the substrate gets converted into the product with less hassle. So basically, enzymes reduce the activation energy of a reaction. So the answer here is false. Uh, yeah. The next question is, in a reaction series wherein an energetically favorable reaction is coupled with an energetically unfavorable one is known as option A, energetically coupled reaction, option B, energetically independent reaction, option C, oxidation. Sorry. Option C, oxidation reduction reaction. Option D, none of the above. Uh, yeah. So you can take a couple of minutes and then raise your hand, and then we'll talk about each of the three different reactions and decide which is the right reaction. Yeah, Kritika, we can start with you. You can define what is an oxidation reduction reaction.
So basically, oxidation reduction reaction is known as redox reaction, in which one is oxidizing and another is reducing simultaneously. So, if energetically favorable means that is feasibility we are talking about, right? So delta G should be negative. And when we talk about the energetically unfavorable, that is delta G positive. So in case of oxidation reduction reaction, I think this is not the option for this.
Okay, okay. Um, yeah. And Surender, you can talk about what energetically independent reaction is.
Independent reactions are which are happening without influencing each other.
Huh.
They
So,
both may be thermodynamically favorable. Okay, this is the only case. Independent. Both are the independent reaction. Both are happening independently. So both must be having the though both must be thermodynamically favorable.
right. And Ayush, you can Ayush, you can talk about what energetically coupled reactions are.
So generally, the energetic couple reaction is like coupling allows the overall process to proceed energy in a favorable condition.
Y-
Reaction मतलब
Yeah. Can you come up with any? Do you have any reaction in your mind that you have learned previously where the reaction is coupled? Any idea? Sorry. Ayush, 1 minute,
so
Kritika.
I'm not able to think right now.
Okay. Like, uh, yeah. So, like Kritika had mentioned about oxidation reduction reaction, which is basically redox reaction and transfer of electrons happens, and one species gets oxidized, the other species gets reduced. And energetically independent reactions are those uh, reactions where uh, uh, because these reactions are feasible thermodynamically, they do not need an external source which would allow them to improve their feasibility. Whereas energetically coupled reactions are where you have a reaction whose delta G is not very favorable. So what happens is this is coupled with another reaction where a high energy molecule like is ATP's hydrolysis is coupled with this energies reaction. So what happens when ATP is hydrolyzed? It gives ADP plus for one inorganic phosphate. This releases a lot of delta G, more delta G or more free energy that is sufficient for the previously unfavorable reaction to become favorable. So this is called as energetically coupled reaction. Yeah, Kritika, you can share your thoughts about this.
Yes, so you were asking an example, no? So like in ETC, we have ATP
Yes,
synthesis. That can be the example of energetically coupled reaction.
ATP synthesis in electron transport chain.
Yes, sir.
So that happens in the presence of proton motive force, right? You have
Yes.
you have a gradient, which is a gradient of hydrogen ions. So you have a lot of hydrogen ions outside compared to inside. So what happens when these hydrogen ions traverse the membrane through this ATP synthase? you have production of ATP. That is what you were trying to say, right?
Yes, sir.
Okay, technically we can't call that an energetically coupled reaction because we are there, ATP is being produced. What has been categorized as energetically coupled reaction is a case where you have a reaction where A forms C, A plus B forms C, but the delta G for product formation of C is not positive, it is greater than zero. Now, what can be done about this? You couple this with hydrolysis of ATP. So, ATP gets broken down to form ADP plus phosphate. Now, a delta G is released for this particular reaction, which is coupled with the previously unfavorable reaction. So, what you had cited as an example might not be suitable here, but there are
Okay, sir.
cases in
Got
the
it.
literature which talk about energetically coupled reaction. Yeah, Surendra, you were about to say something. You can share your thoughts.
Energetically coupled reaction.
Huh.
As we discussed, one reaction having the delta G minus and other having the positive. Delta G
Yeah,
is positive.
yeah,
But when you combine both delta G,
huh.
overall delta G will be negative.
Yeah,
I got it. But I'm
yeah.
still getting confusion why oxidation reduction reactions are not considered under this category. Because if you consider the example of any dehydrogenase enzyme,
Okay,
okay, one is reduced, other is oxidized.
okay,
If, for example, glyceraldehyde 3-phosphate dehydrogenase is there, it
okay.
converts the glyceraldehyde 3-phosphate to 3-phosphoglycerate,
DHAP. Yeah, yeah. Huh,
and
huh,
NADH is produced.
huh.
If we are not provided with NAD plus, then how this reaction is possible? If both
So
are coupled,
there, these are reducing equivalents. They don't provide you delta G. Reducing equivalents take charge. So chemically, you need a species to take up protons or to give up protons. That is what you had defined in that particular reaction, if I'm not wrong. But
yeah,
the
yeah.
energy that comes is what is asked in the question here. They don't talk about charge transfer or proton transfer. Only when they talk about this charge transfer or proton transfer, then you can call that particular reaction that way. Was it clear?
Yeah, yeah, yeah.
Okay. Huh. The next question is nutrient uptake by the cell has
Uh,
prob. Yeah, sorry.
can,
Yeah,
uh,
yeah, yeah. Should I go back?
so, yeah, yeah.
One. Huh. Yeah.
Uh,
Your doubt is?
for energetically coupled reaction, there should be involvement of energy in the form of ATP.
I have read about cases of ATP. I think cases of other triphosphates like GTP also do exist. I can check and I can
Uh,
get to you.
but sym-porter transporters are there, na? Sym-porters,
Yeah.
where one molecule goes above the against their concentration gradient, and another will go down their concentration gradient, and they're coupled. And there is no involvement of ATP, GTP there. But we call them energetically coupled, na? Thermodynamically favorable. Antiporter or
Antiporter.
sym-porter.
Antiporter. Where two. Okay, I understand what you're trying to say. You are saying in a membrane when the transporter is there and two different solutes go opposite each other, even if one of them have high delta G, the transport for one of them have very good delta G, the others transport is still facilitated. That is what you're trying to say, right? Surinder, did I get it right?
Yeah.
Huh.
My question.
So I can check and I can let you know. I am not really sure if, although there you have the transport coupled between the two solutes, across the membrane. I'm still not sure if you can categorize that as an energetically coupled reaction. I can, I Well, I'll make a note and we'll try discussing it next time.
Okay. Thank you.
Yeah, yeah. 1 minute. I'm just making a note of it. Yeah, the next question is nutrient uptake by the cell has properties like option A, non-specificity of the uptake process, option B, uptake of the molecule down or against the concentration gradient, option C, nutrient molecules can pass through any cellular membrane, option D, none of the above. You can take a couple of minutes and then we'll get back to this question. Yeah, Surinder, you can talk about whether the molecules that are being taken up are specific or not specific. How does the nutrient uptake happen?
Uh, it is specific because so many molecules are present in the environment of a cell.
Right,
But cell uptake only those that are required for its growth, for its maintaining the homeostasis.
right.
Nothing else uptaken by the cell. That's why I think it is a incorrect.
So that option is incorrect. Okay. Ayush, you can say whether nutrient molecules can pass through the cellular membrane or
So, as we know, nutrient can uptake or क्या बोलते हैं uptake the molecule down or against the concentration gradient because like uh, in passive diffusion the molecule uptake is from lower concentration to from the higher concentration to lower concentration, but in active transport it can move against the concentration gradient but it needs the ATP energy for uh, for the uptake of the
Okay. Kritika, you can talk about the cellular membrane first and then try to say whether all nutrient molecules can pass through the cellular membrane or not.
So when we talk about the cellular membranes, so these are like made up of phospholipids layer by layer, and outer one is hydrophilic and the inner tails are hydrophobic. So those molecules which are hydrophobic, like those are lipid like molecules, they can easily pass through the membrane. But when we talk about the hydrophilic, like we have water soluble vitamins, vitamin B and C, these are hydrophilic, so they will not pass easily through the cell membrane.
Okay.
Yes, sir.
So you're saying that nutrient molecules cannot pass through any cell membrane.
Not specific, like it depends upon their polarity, then
Huh.
only they can pass through the cellular membrane. It's
So,
not
so we
true
can't
that
make a
every nutrient can pass
generalization.
through them easily.
Huh. So we can't make a generalization that all nutrient molecules can pass through. Yeah.
Yes.
So based on this, option A and option C are wrong. So option B is only right, which is the uptake of the molecule can happen against or down the concentration gradient. Good. Uh, the next question is select the compound that is considered as a low energy indicator of the cell. We can take a couple of minutes and then we can discuss this. Yeah, does anyone want to answer why option? Uh, okay, wait. We'll first see why triphosphates are given as energy indicators of the cell. Yeah, Surendra, you can try to answer why option C or D could be the indicator or could not be the indicator.
The COD option.
C or D option. It's up to you. You can even talk about both the options if you want, if you are aware, or we can discuss. Yeah.
Because A and D both are indicator of the high energy because they are having the three phosphodiester bonds,
Huh.
phosphoanhydride bonds. Okay, and breakdown of each in removal of each inorganic phosphate from the either GTP or CTP, it releases some energy. Okay,
Right.
so there is some there are three phosphates. So when we remove three phosphate, it releases more energy
Right.
as compared to if we remove the two phosphate or one phosphate.
Okay.
That's
Ayush,
why I think COD they are the high energy.
high energy indicator. Okay. Yeah. Ayush,
yes sir.
do you know the difference between ATP and AMP?
Yes sir.
Okay.
Sir,
Let's say,
adeno triphosphate or adeno monophosphate.
okay. So, so, sorry. So let's say within a cell, we have 75% AMP and 25% ATP. So what could this mean to a cell? Initially, we had 100 ATP, but later on, after some time, we have 75 of AMP and 25 of ATP. What could this
So,
mean to a cell?
it could mean that the ATP is converted to AMP and release some energy
Huh,
because ATP is hydrolyzed to AMP and the inorganic phosphate
huh,
and, and release the energy. So, or
oh,
what else?
huh. So this released energy, does it go wasted in the cell, or maybe it is useful for a function?
So,
So you said ATP is converted to AMP and inorganic phosphate, and some energy is released. So what I asked is, the energy that is released, is it doing some useful work inside the cell, or is it getting wasted?
so So
Okay.
generally,
Huh, yeah, sorry, you can.
the ATP used in multiple uh, processes like glycolysis and the various cellular activities. I'm not able to recall, but sir, is generally used as a initiator, sir. Another like
Okay, so just to give some clarity, ATP is broken down to give ADP and phosphate, and ADP is broken down to give AMP and another inorganic phosphate. And all of this happens only when the cell requires energy for a particular process. So whatever energy that we get out of ATP hydrolysis is utilized. So the cell does not have any more energy. So having more amount of AMP in the cell would mean that it had ATP, but that energy was utilized to give it into AMP, which means that now there is low energy on the cell. So the right answer is option B, AMP. Was that clear?
So you're not audible.
Oh, sorry. Uh, am I audible now?
Yes, sir.
Yeah. So what I was saying is ATP is a high energy source. When it's getting hydrolyzed, it forms ADP and inorganic phosphate. When ADP is getting hydrolyzed, it forms AMP and inorganic phosphate. So what happens in this process is that some energy is being released. This energy is not being wasted, but energy is utilized for the cell's cellular role. Some functions are being performed with this energy. Now, what happens when a lot of ATP is converted into a lot of AMP is most of the cellular cells energy is being converted, meaning it goes low on energy as ATP gets converted to AMP. So the low energy indicator of the cell is AMP. Was it clear?
Yes, sir.
Okay. Yeah. The next question is, the entry of pyruvate into TCA cycle is inhibited by the presence of high cellular concentration of option A pyruvate, option B NADH, option C coenzyme A, option E AMP. One can think about the TCA cycle, or I can even try to show that, and then we can discuss how entry of pyruvate into TCA cycle is inhibited. Yeah. So, Surinder, you can start explaining. If you want, I can share a picture of the TCA cycle, depending on your explanation, or even you can share if you have. I can allow you to present. Would you want me to share a picture of the TCA cycle?
Uh, um, I try to without this.
Okay. Yeah, you can start by explaining.
So entry of the pyruvate into TCA cycle.
Yeah. How is it inhibited
So pyruvate,
when?
huh,
Huh.
pyruvate enters into the TCA cycle. First, it converts to the acetyl CoA.
Okay.
Pyruvate convert to the acetyl CoA by the help of PDH complex.
Right.
So pyruvate is the substrate.
Right.
Okay, and coenzyme A is also the substrate for this reaction.
Okay.
For the conversion of pyruvate to acetyl CoA, pyruvate and coenzyme A, they are the substrate. And substrate never inhibit their enzymes. Only product inhibit by the feedback inhibition.
Ah.
Motto of the TCA cycle is to produce the more reducing equivalents as well as the energy, ATP in the form of ATP or GTP.
Okay.
Okay, and AMP is the indicator of the low energy.
Huh.
Okay, so AMP is also not going to inhibit because AMP is the indicator of low energy, more energy required by the cell.
Right.
Okay,
Right.
and NADH, it is the product of the TCA cycle.
Right.
Okay,
Right.
if more NADH are there, I think it is going to inhibit the entry of the pyruvate into the TC cycle. NADH will inhibit. This is from my side.
Okay. Kritika? Yeah. Kritika? Ayush?
Yes, sir.
Yeah.
Sir, I also think, sir, the higher concentration of NADH will inhibit the entry of pyruvate into TCA sector because as the pyruvate converted into acetyl CoA, so it needed coenzyme A plus NAD plus. So if then it get protonated, so it releases the carbon dioxide plus NADH2. So NADH plus uh, proton
NADH plus H plus. Okay.
any higher concentration, so the it will not allow the entry of pyruvates.
Okay. So, um, yeah, good. Um, so, as mentioned by Surender, the answer is option B. Yeah. Okay. Yeah. So, now that we are done with the sample questions for this week, we will look at a topic of discussion that we had last week. So, to remind you all, Surendra had asked how facilitated diffusion can be categorized as active or not. So, what facilitated diffusion means? There are compounds that are not very soluble in the plasma membrane that are transported at an extremely slow rate. When these compounds or molecules are being transported, they use carrier molecules like proteins that are within the membrane. But it is mentioned clearly that it requires no energy and can occur only in the downhill direction of the concentration gradient, which is why it's called facilitated diffusion. Uh, this is just to clarify the last meeting's conversation, Surendra.
Actually, my question was, it was the question, active transport related to active transport, whether it
Yeah,
requires a carrier or not.
whether
It was
facilitated diffusion can be classified as active transport because facilitated diffusion involves a carrier molecule. That was the question, right?
no, no. Question was, there was a question, active transport is
Yeah.
carrier mediated. Option A was the carrier mediated. Option B was something else, and option C die. So D and E was the correct. You have not considered A.
I'll open option. I'll open the that slide. Let me give me a minute.
First,
This was assignment zero, right? 1 minute.
yeah, assignment J.
Assignment zero, last question, sir.
One. Let me present my screen. Yeah. So here is the question. I hope my screen is visible. So this is the question. Active transport of nutrients across the biological membrane is characterized to be option A, carrier mediated, option B, occurs from high concentration to low concentration, option C, requires energy, option D, occurs against the concentration gradient, option E, sorry, option E, both A and C, option F, both C and D. So out of these options, mm, because you're looking for active transport, it means that it does not occur with high concentration or occur from higher concentration to lower concentration. It means that it requires energy and it against the concentration gradient. So the doubt that was raised is whether carrier mediated transport can be called as active transport or not. This was the question, Surender.
Yeah, this was the question.
Yeah. So they clearly say that. Sorry. They clearly say uh, that these two options that it requires energy and against the concentration gradient are right. But we were doubtful if carrier mediated, which involves a protein for a solute to move from one side of the membrane to another, can be called as active transport or not. So
Mhm.
here in lecture 8, they discuss about transport using carrier molecules. They say that it requires no energy expenditure and occurs only in the downhill direction. So technically, as per this lecture, it what we had discussed last week is right. Carrier mediated diffusion cannot be categorized as active transport. Was that clear?
Actually, facilitated diffusion is always against down the concentration gradient, and it requires some carrier.
Okay.
And active
Uh,
transport is against the concentration gradient, but it also requires a carrier. So, in my opinion, A should be considered as a correct answer,
okay.
because without carrier, active transport is not possible.
No, with energy, with just the supply of energy, but not. Okay, I'll try to get an example, maybe. Example of active transport without carrier. Let me check.
Mm.
So, just to clarify, when we say that something is a carrier, we mean that it is a protein that necessarily uh, helps a solute molecule move inside and not allows exactly. Al- Membrane proteins are of many kinds, and allows some transporters help the protein to move inside. The sodium potassium pump case, if you see, they bind and allow the antiport movement of sodium potassium. So in those cases, we can call it carrier. But in some cases, energy is needed so that transporters are open, then solutes can enter. So this is the active transport that I think is being discussed in the question here. But I'll try to get you an example of active transport without carrier. But
Yeah, yeah.
it will have a protein involved. What I'm trying to say is there could be proteins involved in active transport, but
Yeah.
these proteins will not necessarily help your solute to move. They might be involved in the process because they block the membrane and prevent your solute. So they basically allow your solute to move from one side of the membrane to the other, but they do not necessarily like handhold and allow the like help the solute pass through the membrane. That is what I wanted to clarify. But yeah, I'll uh, I'll see. I'll try to get an example. Other than this, there were a couple of questions raised by Surinder in the forum. Excuse me, sorry. So uh, I'll share the Google Forums link. Please do check the Google Forum and join the Google Forum and post your doubts there, like Surinder did, so that that can also be brought for discussion uh, when we do this live session, and it's beneficial for all of us. Yeah. So he had raised two questions, both from the same video. Uh, so the let me go to the forum. Yeah. Okay. I haven't seen this third question. I'll check that. I had prepared the slides a few days back. So I'll answer the third question if time permits today. Otherwise, we can do it next week. So the first, so the first question is, uh, yeah, in the video, as per you say at this particular time point, the professor says that. there is glycogen present in E. coli cell, and that it is contributing to LPS
polysaccharide, or within the
or
cell
in
wall,
the cell
the
wall.
different type of amino acids and nucleotides, other amino acids.
So in this video, if you see, he says that glycogen contributes to lipopolysaccharides and the cell wall. So one one thing that we can consider is that uh, glycogen is a sugar source, although it does not directly contribute to the polysaccharides that form LPS and NAG and NAM that contribute to cell wall. Maybe uh, the professor might have meant that glycogen derived products are used in the formation of NAG and NAM. But to clarify, glycogen is not present as glycogen in lipopolysaccharides or in the cell wall. Although there are reports stating that in yeast, which is Saccharomyces cerevisiae, glycogen is covalently linked to the cell wall. And I have put a link to that in the slide, which will be later shared with you. Uh, so to clarify, it is not present in bacterial cell wall, but there are cases where glycogen was observed in the cell wall as such. Was it clear, Surender?
Yeah, yeah, yeah.
Yeah. And the next question was energy. Yeah, the difference between energy conserving reactions and sorry, catabolism. So, so what people consider as energy conserving reaction and catabolism is different. Like in the video, the professor says the professor doesn't say that all energy conserving reactions are catabolism. This is because energy can be conserved in two interchangeable forms as energy rich bonds or as electrochemical gradients over membranes. So in the video lectures itself, we have discussed something called as proton motor force, where we see a gradient, a electrochemical gradient of hydrogen ions on one side of the membrane. So when there is a higher concentration of hydrogen ions, and when it moves from higher concentration to lower concentration, that in a way activates ATP synthase to produce ATP. So there are two different kind of reactions that can actually conserve energy. One is through the bonds that we see in phosphates, the other is through these electrochemical gradients. So, because of these two different separate entities, we don't always call breakdown, which leads breakdown of energy rich bonds or conservation of energy rich bonds as uh, we don't always categorize catabolism as energy conserving. There are other reactions also in the electrochemical gradients that contribute to energy conservation. Uh, was it clear, Surender?
Yes, yes.
Yeah, okay. Since we have time, I look at the third question, but uh, I'll see what exactly is the question. I haven't seen it earlier. Okay, so membrane thickness is measured in meter.
By the concentration in the abiotic phase and concentration in the cytosol. So if we know,
So although they have given it, so these are the SI units, Surender. So although they have given it in meters, it will be because the membrane thickness will be in nanometers, a few hundred nanometers. You can consider it as 10 power minus 9 meters. So that is not a thing that we need to
Okay,
be exactly bothered about. It it
okay,
should be accounted for mathematically.
okay.
Yeah. So with this, we were able to cover the questions and the doubts related to week two's lectures. And if you have any more doubts, we can discuss that, or we can finish the meeting here today.
What is ED pathway?
Uh, Entner Dodornoff pathway, right?
ED pentose phosphate pathway. It originates from the glucose 6 phosphate.
So there are three glycolysis routes. One is EMP pathway, which
Okay.
is conversion from this is the normal conversion, which leads to production of pyruvate from hexo 6 phosphate towards uh, phosphoenol pyruvate to pyruvate. The second one is pentose phosphate pathway. Here, what happens is hexo 6 phosphate gets converted to phosphogluconate, and that gets converted to different kind of sugars like pentose sugars, 4 carbon sugars, and 7 carbon sugars, which ultimately gives to glyceraldehyde 3 phosphate. And third is the ED pathway, which is predominantly seen in pseudomonas. So here what happens is similar to the pentose phosphate pathway, we have conversion from hexo-6 phosphate to phosphogluconate, which gives rise to 4 carbon, 5 carbon, and 7 carbon sugar. Then this gives rise to glyceraldehyde 3 phosphate, which gives rise to phosphoenol pyruvate. Apart from this, these 4 carbon, 5 carbon, 7 carbon sugars can also give rise to pyruvate. So basically, all of these are interlinked, and a part of the pathway is given a specific name. So, did that answer your question, Surendra?
Yeah.
Okay. Yeah, Kritika, you had raised your hand.
Sir, is it true that during fermentation, NAD plus and H plus in concentration increases?
Uh, during fermentation, I, I can check that and tell you. I am not particularly aware of the concentrations, but if the question can be a bit more specific, I can try to search and I can tell you. I'm not very sure.
Sir, it's in our assignment two. There's a question regarding the fermentation, and they have asked like which is true about the fermentation. So there were four options basically. First is like organic carbon as the source of electron,
Huh.
something like that, sir. And um, fourth condition. So it's an assignment two question, sir. I don't remember it properly
Uh,
actually.
Kritika, in my opinion, NADH concentration is not going to increase because for fermentation, pyruvate to lactate formation, pyruvate to ethanol formation, NADH is used. Okay, so it is not going to increase, in my opinion. Okay,
But it's breakdown in NAD plus and H plus. If the rate of fermentation is increasing, then it will not increase.
NADH, it is asking for the reducing equivalent, I think. Huh? In question, reducing equivalent. They are actually, I have
I
not gone through the question. Okay.
even I haven't checked the assignment, but Kritika, you can post this. Okay, because it's an assignment problem, you can't technically post this as a question in the group, but you can read on it, and I'll try to discuss this
Yes, sir. The
in the next
question
group.
is
Ha,
like in the fermentation process,
ha,
first is the organic carbon act as an electron source.
ha.
Second is oxidative phosphorylation serve as a platform for energy production. Third is both O2 and NAD plus act as electron acceptors. And fourth is the concentration of NADH and H plus increases. So we have to tell which statements are true. So like I'm sure that oxidative phosphorylation it's not the like platform for energy production. So this one is false.
Oxygen
Uh,
also
we
needed there.
we can't really discuss the assignment questions in this forum. We can, because these are supposed to be evaluated, but but once you submit the assignments, maybe we can discuss. By then, I'll try to read up on this and I'll get back to you. That should be fine, I
Okay, sir.
guess.
No issue, sir.
Okay. Thank you. Thank you for joining. We'll meet
Thank you.
next Monday at 6 to 8. Thank you, Surinder, Kritika, and Ayush.
Thank you.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.